This lesson is going to go over the steps of protein synthesis. And so there are two phases, and the first is transcription and the second is translation. So first off, in transcription, this is going to occur in the nucleus. And so an enzyme is going to bind to our double-stranded DNA and unzip it into single strands. And then we have an RNA polymerase molecule that's going to come in, read the single-stranded DNA, and create an mRNA strand by base pairing with that DNA strand. So if we look at an example here, we'll make a DNA and an mRNA strand. So for DNA, we'll just throw out some letters, just a random sequence here. When we look at this in our DNA, adenine always pairs with thymine, and then in RNA we have no thymines because it gets replaced with a uracil. So A always pairs with U. We have no T's present in our RNA strands. And then in both DNA and RNA, we always have a C pair with a G. That won't change. So if we look at this, we have our enzyme that made our single-stranded DNA, and then our RNA polymerase is going to come, and the mRNA strand that it's going to create after it reads this is going to be AUG, and this happens to be our start codon, and then GGA, and this is just a regular codon. And so when we do transcription, we're going from DNA to mRNA. And one important note is U replaces T in RNA. And then from here we go to translation, which is going to occur in the cytoplasm. And so the mRNA strand that was created is going to bind to a ribosome because that's the site of protein synthesis. And then a transfer RNA strand is going to come in with its anticodon. And this is going to continue, and the ribosome is just going to keep moving from codon to codon, bringing in the specific anticodon. And each of these anticodons codes for a specific amino acid, and each of these amino acids will be linked by peptide bonds, and this process will keep repeating until we reach a stop codon, and then once we hit a stop codon, the polypeptide will be released. And so again, if we go through an example like we did before, we have our mRNA strand, which is just AUG, and then we have GGA. The transfer RNA strand that's going to result from this is the anticodon will be UAC, and this code is going to code for a specific amino acid. And there are charts that you can look up these anticodons to determine which amino acid will result. So this one is going to be CCU for our anticodon, and then we're going to get a specific, we're going to get proline for our amino acid, and these will be joined by the peptide bonds. And then this is our overall direction of protein synthesis. So this is the direction that the ribosome will go. It starts at our start codon and just keeps reading until we hit a stop codon like we said before. and so then the polypeptide will be released. So at the bottom we have one final little kind of a summary of everything. We start out with our DNA, and then we go to an mRNA molecule, and we have it's separated by individual codons, and then we have our transfer RNA, which comes into play with the anticodons, which are linked with specific amino acids. And once we get a chain of amino acids, this is what forms our protein. And so the way that we're able to get such a huge variety of different proteins in our body to perform all of these different functions is because of the different amino acid sequences. And there are actually only 20 different amino acids total, but because they can arrange in such a variety of ways, where different lengths of chains as well as in different orders, we're able to create a huge variety of different proteins.